Hi, Assalamualaikum and salam sejahtera. My name is Muhammad Hafizi bin Ibrahim, metric card number 2021878412. I am from AT2203A, and today I would like to present AGR514 video presentation titled Disease in Oil Palm in the focus of the Basal Stem Rot. So this is the introduction. The Basal Stem Rot, or BSR, is a fatal fungal known as Ganoderma Boninense disease of oil palm plantation and has a significant impact on the production of palm oil in Malaysia. Because there is no effective treatment to control this disease, early detection of BSR is vital for sustainable disease management. The limitations of visual detection have led to an interest in the development of spectroscopically based detection techniques for rapid diagnosis of this disease. The aim of this video presentation was to elaborate about the causal disease symptoms, the sign of pathogens, how Ganoderma affect the function of oil palm tree, and the disease control. So we go for the causal disease symptoms. The young palms, the lower leaves become yellow or develop yellow or green irregular spots and patches called a mottle. They bend downwards and then begins to rot prematurely. The unfolded leaves become yellow, shorter than normal, and sometimes develop rots at the tips. Later, growth slows, palms become pale, and spear leaf remain unopened. Symptoms of mature palms are similar. They also show multiple unopened spear leaves and pale leaves. Internally, dry rots occur in the stem and bole, and dry powdery rots occur in the roots. Young palms will die within 6 to 12 months of first symptoms, whereas mature palms may survive for 2 to 3 years or longer. Okay, we go for the sign of pathogens of the basal stem rot. DSI number first for mature palm stands for the disease severity index, where we can see here mild or moderate infected palm with the presence of white mycelium of fruiting body, like a small white button or bracket shape form. This stage, palm not showing or showing foliar symptoms, less than 50%, and slightly stem rotting, around 10 to 30% at the base. And the confirmed presence of Ganoderma fungus using early detection methods like GSM or PCR DNA. The number two level of disease severity index, the severe or very severe or death infected palm. Then we can find the presence of white mycelium of fruiting body like small white button or bracket shape form. The palm death and collapse showing severe foliar symptoms and stem rotting at the base. Confirmed presence of Ganoderma fungus using early detection, same as the DSI1 methods, the same using the GSM or PCR DNA. So this is the epidemiology disease spread with the mycelium contact between healthy roots with diseased tissues left buried in soil. We can see here from the diseased palm, the roots contact with the diseased tissue, and it will infect the healthy root with the healthy palm. And the source is from the Ganoderma inoculum. Okay, the next topic is how Ganoderma affect the function of oil palm tree. The symptom of BSR or basal stem rot are decay of the bottom of the stem from where basidiocarps emerge, and sometimes also decay in the roots. Stem rotting restricts the uptake of the water and nutrients from the fronds and causing chlorosis. When the disease is more advanced, the older fronds wilt and hang down to form a skirt around the trunk. Other observable symptoms are flattening of the crown and sepia leaves that have not opened. In the most severe cases, the stem might even fracture, and Ganoderma can cause mortality by damaging the trunk xylem tissue through production of enzymes that degrade lignin components, which are then consumed by the fungus. This interrupts the distribution of water and nutrients in the tree and results in the appearance of specific symptoms such as yellowing and necrotic leaves, unopened spears, small canopy, and skirt shape of the palm crown. So about the Ganoderma disease control, we have the chart here. We have quite lots of the Ganoderma control and management. But in this presentation, I would like to mention about a few control and management of Ganoderma disease. Number one is sanitation by removal or deboling of infected palm. After we did the census, then we got the DSI 1 or 2. Then we pushing the infected palm using backhoe excavator. Then we do excavating stem and root masses from 1.5 to meter length and times by 1.5 width or times by 1 meter depth. Then after that, we refilling with the nearby soil. We chip the trunk, stem and root masses much less than 10 centimeter thickness, and we resupply the palm in the field if the existing stem is less than 10 years old. Number two, we use the fumigant Desomet with the stem treatment for eradicating of Ganoderma inoculum. And as I read in the field study and based on my research, Ganoderma inoculum significantly reduced when infected stems treated with Desomet. And Desomet moves into the stem and cause death to the Ganoderma fungus, therefore reducing the spread of Ganoderma disease within the oil palm plantation. So the steps like we apply Desomet or Basamid at 500 grams per stem, and we spray the water with 250 milliliter and cover with a polythene bag or with soil. After we did the BSR census until the level 2. This one number 2. So number 3, we using fungicide for the infected standing palms with no foliar symptoms, and we call it the productive palm. We call it a curative treatment and prolonging life of infected palm. And there are 2 fungicides that usually we use, and hexaconazole and tetraconazole. And as today, we completed the 3 field trials. Namely is Gamat, Johor, in Sepang, Selangor, and Teluk Intan Perak. And in these 3 trials, showed we treated with the hexaconazole using hand knock injector significantly lower dead palms compared to untreated. And an average of 74.4% from the 3 trials of infected palm treated with hexaconazole still alive and producing fruit bunches, and none of untreated palm. So we can see here the tri injection with fungicide hexaconazole on Ganoderma infected palm. So the step is after we did the census, we remove the old front buds using chisel or chainsaw. So we do the drilling to holes into the trunk or stem at the base using motorized drill. Then we prepare the fungicide solution. Then we inject the fungicide solution into the drilling hole with a total of 6 liter with a 3 liter per hole using injectant attached with motorized knapsack sprayer. And after that, finish the fungicide solution into drilling a hole. We do the spraying 1 liter fungicide solution onto the stem or trunk at the base. So the number 4, the strategies to control Ganoderma disease at replanting. First, we do the excavating, deboling of boles, stems, and root masses of all old palms. We do the ploughing along the new planting row, and we planting new palm along the ploughing area. So as the long-term control strategies, as a preventative treatment, we can find in the market is the commercial products such as Gano AF biofertilizer using the Hendersonia Gano EF1. This one suggested and studied by the Dr. Idris from MPOB. Number 2 is MBO, also recommended by the Dr. Idris. And the last one is Gano Care, organic and OC special fertilizer, also recommended as per study by Dr. Idris as a preventative treatment, long-term control strategies. And lastly, for conclusions, what I can say is among oil palm disease, BSR or Ganoderma disease is the most widely studied and knowledge available. And the Ganoderma disease poses a major threat to oil palm industry in the near future if no control measure is implemented. MPOB has transferred several technologies on controlling and managing of Ganoderma disease in existing planting and at replanting. Preventive products are available in the market. And last one is some prevention and curative treatments developed are being adapted and implemented by oil palm industry and smallholder. Thank you very much.